Hi, Aaron.
Hey, RP, how are you?
Good. How are you doing?
Good.
Thanks for
Thanks
organizing
for.
tonight.
Oh yeah, it's easy. It's fun. Thanks for agreeing to give a talk.
My pleasure.
I think Mark Freeman volunteered you. Actually, all
Yeah,
volunteered
he's
all of
he's
you.
volunteered me in lots of great things.
Nice. Cool. You want to give your slides a test?
Sure.
Oh, let me. I think I'm going to make you a co-host, or
Okay.
um, that might give you a little bit more powers. But I think
How's that?
looks great.
Okay, cool. All righty.
Cool. How's everything up there, Portland?
It's nice. It's really nice. Now, you spend time at the Vollum, right?
Yeah, for a while I was coming up there like every couple years. Like Gary Westbrook would invite me to teach in his class, and I just love coming to Portland. But I never spent like any formal time there.
Hey, Aaron, how are you?
Hi, Holly, how are you doing?
Good.
Thanks for joining.
Yeah, of course. My pleasure. Let's see. So do you want to check and make sure this all works?
Yes, please.
Okie doke.
Looks good.
Right?
Yep, looks great.
What's your doggy's name?
Oh, that's Archie.
Archie.
Yeah, she's a Labradoodle.
Oh, really?
Yeah.
She's not so fuzzy for a Labrador.
She is. Archie, come. Let's see. Yeah, she's kind of fuzzy.
Yeah.
She looks like she's kind of been there, done that, the Neuro Zoom.
Yeah, she's always there. So I'm in Shanghai this year, and we brought her all the way to Shanghai with us. So that was kind of a challenge, but she's having fun.
Did she have to go through quarantine?
No, people do, dogs don't. But she had to go she had to go through Germany. Like Lufthansa is the only carrier that will bring dog. Anyway, it's complicated. But she went to Germany, and then Germany to Shanghai.
Wow. Yeah. Will she have to go through quarantine on the way back?
Not that I know of.
Yeah, that's cool. No, I just thought that was part of the.
We had to do a lot of tests to get certifications and rabies vaccines and all that other stuff, and then get everything. All these, we hired there's like a dog shipping agent, so we hired like a dog shipping agent and to do everything.
Well, she looks he looks pretty well adjusted.
Yeah. She's been here. We've been here about 4 months, so yeah. So I my wife was here earlier, so we shipped her first, and then I got her at the airport, and then I brought the kids. And we just came. And we did the quarantine.
How long are you in Shanghai?
TBD, but I'm like on a sabbatical.
Oh, cool.
Yeah.
Hey, Holly.
Hey, John, how are you?
Good to see you.
This isn't a brain meeting, is it?
No,
Everything's a brain meeting.
This is my other life. The life of an actual scientist, maybe. I don't know.
Cool. Yeah, that's nice.
John always has the best Zoom cameras.
It's all the other stuff we got to deal with.
Yeah.
Well, I'm looking forward to both talks today.
Hey, John, how are you?
Good. How are you?
Good. I remember fondly meeting you during my PhD interviews. That was a long time ago.
Oh my God. Did we accept you, I hope?
I think you did.
You didn't come though.
No, I stayed on the East Coast with my family, but
Hey, it worked out
I had a
well.
great time.
It worked out well. Can't argue with the results.
Hey, it's long. Yeah. Yeah, RP, Holly. Hey,
How are you?
RP. Oh, it's been a while.
Yeah. I can't even remember when we saw each other.
It's time I left US 2009. It's our course from Harvard.
Huh? Yeah.
Yeah. Oh, it's long. I switched over the Neuro Zoom webpage. I don't know.
Okay.
I don't know if anyone. It was on Google, and then Google's always a pain to log on in here, so I just switched it over to same website address, but now it should be easier to
Okay.
access.
Is the Google one still working?
It is, but I'm not gonna update it. But it's everything. It's like it's identical. Like we can.
So when we type in neurosome.bio, okay.
Yeah, it'll still go. Yeah. So it's just I just added like a black brick thing, but everything else is really like identical. The list, everything.
Oh, cool. Yeah.
But it just goes right there.
Yeah. Well, when are you gonna switch back to the this normal time?
Oh, November 8th. Yeah, I'll start making announcements. November 9th, 8th, 9th. Yeah.
Okay.
What's going to happen?
We switched to standard time, and China basically it means nothing to us, but in China the talks will be at 9 a.m. instead of 8 a.m.
Oh, that's not better.
Yeah, it's 1 hour late.
Yeah.
Mhm.
So that's good.
But all of China is in one time zone, is that correct?
Yeah.
I'm trying to convince them to change that, but they're not listening to me.
They're not.
the people in the west part has a long night. So they had like a dinner at very late time because
I want to take my dog for a walk at like 5:30 p.m., but it's already dark here.
Yeah.
So that's the only suggestion I have.
We had summertime before actually. Yeah, it's been a while. When I was in high school somewhere, we had summertime for a while. Then people said, "Oh, okay, this is such a pain as to switch time over again. Just stop doing it." Yeah, so we just quick the summertime policy.
Okay.
Yeah.
But like in some parts, in the west and the north, it's really tough.
Yeah, it's just, in the west part, like Kunming, the Yunnan part, they have usually have dinner at like 8 p.m. because it's dark. It's very fun to visit there.
Yeah, I think I'm gonna go there in December.
Cool. Yeah, it's the best time to visit, and it's like San Diego, in China. It's always spring, and it's never hot summer and winter. It's best place.
Yeah, it's known as the Garden City, isn't it?
Yeah, exactly. It could be.
Oh, nice.
Because they have roses providing the whole China because it's growing through the 4 seasons. Yeah. Also, it has a very nice food. And it's weird. It's not, but it has all kinds of spicy and mushrooms because the weather is so good. So you have all kinds of mushrooms, including some mushrooms supposed to containing some you know, weird substance which is supposed to cause some illusional. And people always suggesting you always cook the mushroom very thorough, like never eat anything halfway. So yeah, otherwise gonna have some illusional, testing some schizophrenia-like symptoms.
Inactivated.
Oh yeah. So every year you have some people like got toxic and eating mushrooms.
I think it's the home of the whole like Chinese voodoo and psychedelic
Oh yeah, exactly.
experimentation. So
yeah, I bet they gotta extract some interesting biological substance from there.
you should work on
I
it.
think yeah. How do you like California? Jing?
Oh, I like it.
Jing?
I didn't know I was a closeted Californian all my life, living in the East Coast, and now I'm coming home.
Great.
Yeah, I
Awesome.
instantly became a Californian like within a week of moving to Stanford. I used to think it was better on the East Coast, but it's not.
Little do we know.
Yeah, I went through quite a period of culture shock when I moved from New York to Berkeley because everybody was so happy and I just couldn't understand it. It just-
A lot of mushrooms.
And that's because of mushrooms, Holly?
Oh, no.
I think we're talking about different mushrooms now.
Yeah, maybe.
All right, Zilong, should I get started?
All right, go ahead.
Thanks. Okay, hi everyone. Thanks RP and Holly for joining us today, and thanks everyone else. We have awesome talks also next week. So just to advertise the next week talks, we have Tifei Yuan from Jiao Tong University and my good colleague Jun Ding from Stanford will be giving talks. Lots of talk slots available. So if you want to present your latest research, just send me or Zilong a note, and we'd love to hear what you're up to. Okay, so with that, Zilong, do you want to introduce the first speaker?
Okay, great. It's good. I'm happy to introduce the first speaker today, the RP Saunders. So RP graduated from Swarthmore College, and after that, he went to Harvard, did a PhD with Bernardo Sabtini, where he trained with the hardcore electrophysiologist studying basal ganglia. After PhD, he went to a genetics lab in Harvard, worked with Stephen McCrow, where he used the state-of-the-art genetics combined with viral tracing or combined with single-cell transcriptome sequencing to let us know the interesting connection between all kinds of brain and cellular component during different normal or disease state. After postdoc, RP started his own lab in Vollum, where he continued to use the state-of-the-art a single viral-based single cell combining with single cell sequencing. So today, RP going to talk about the mapping the brain cell organization with single cell single-virion genomics. Welcome, RP. Take it away.
All right, thank you.
All right.
All right, so thank you so much for the kind introduction. It's really a pleasure to join the Neuro Zoom community, and I'm especially excited to be an opening act for Professor Cline. My name is RP Saunders. I'm a new assistant professor at the Vollum Institute at Oregon Health and Science University. And today I'm excited to share with you new viral technology I've developed to study the cell type and molecular properties of mammalian synaptic networks in ways that are systematic, high throughput, and I hope transformative for an understanding of circuit architectures, mechanisms of synapse assembly, and eventually human neurodevelopmental disorders. So I would argue as neuroscientists that we still know very little about the biology of mammalian synaptic networks. Sure, we have methods to catalog brain cell types and map cells' anatomy, but understanding the process through which these networks are constructed from individual cells of particular molecular types and how many instances of these networks are structured to process information is largely unknown. Now, the situation is different in invertebrate systems, of course, largely because of their stereotyped animal-to-animal connectivity. And this helpful property enables genetic personality of individual cells to be tracked across animals, enabling EM-based connectomes of a single tiny brain to be representative and molecular and functional experiments to be performed essentially on the same cell, but in different experimental preparations. In mammals, we're largely stuck analyzing synaptic networks through laborious experiments at the level of cell populations. And by averaging tiny amounts of information across many, many animals, we can then create circuit summaries that look kind of like this, which are largely anecdotal descriptions of cell type specific connectivity, this one from primary visual cortex in mouse. So my lab aims to generate connectivity maps which are single cell resolved, high throughput, and associated with detailed molecular profiles of each cell. And this research aim is challenging because the brain, as we know, is vast and has many hundreds of cell types with very complicated and expansive geometries. So our lab's approach is to engineer neurotropic viruses, which can teach us about synaptic relationships and other complex features of brain cell organization through the way that they infect. For example, going from a messy, intact cortical circuit such as this one to a simplified representation such as this one, in which cell type specific synaptic relationships are quantified and easily discerned. So in addition to developing technology, we're also dedicated to understanding the mechanisms of viral interactions to deepen our understanding of our high throughput data sets, and because we're simply really fascinated with viral dynamics, especially in the context of diverse brain cells. So traditional methods for studying synaptic connections, such as electrophysiology and electron microscopy-based anatomy, are powerful, but they're also limited in critical ways. Such methods are tedious and slow, generally limited to neighboring cells in small volumes, and do not routinely sample molecules from each cell, and so are typically blind to each cell's type and molecular state. Now, in contrast, advances in single-cell genomics enable genome-wide molecular profiling from hundreds of thousands of cells from individual animals and can be processed on a sequencer overnight. And such methods are not limited to mice. They can also be applied to a variety of species and experimental systems. Therefore, when I was a postdoc in Steve McCarroll's lab in the genetics department at Harvard Medical School, I set out to develop a way to encode synaptic partnership information in each cell's RNA, which could then be decoded using single-cell RNA sequencing. So I wanted to develop a system which had the following properties that would be informed by cell type and high throughput at the scale of an entire brain region or even a whole brain, and to get enough connectivity information from a single animal that we could learn about that particular animal's synaptic structures. I wanted the system to be species flexible, not just limited to transgenic mice, and supportive of quantitative and statistical inferences of the networks that we measure. And finally, of course, it should be fast, inexpensive, and straightforward for labs around the world to implement. So the system that we ended up creating is based on the rabies virus pictured here, a virus which has evolved to transit cells using synapses by spreading from the dendrites of a postsynaptic neuron into the axons of that cell's presynaptic partners. Now, to simplify our interpretation of the networks that we reconstruct, we take advantage of a system that was developed by Ian Wickersham and Ed Calloway that restricts the transmission of a virus to a single synapse. And the key here is using a version of the viral genome in which the gene required for the virus to enter cells is missing and has been replaced by GFP. So let me briefly illustrate how this system works. So first, we genetically endow a small number of cells with a special receptor that allows rabies virus to infect only these particular cells. And then in the very same cells, we supply the missing gene in trans required for the virus to spread. And we call these the starter cells. Now, once a starter cell has been infected, the virus replicates and then uses the complemented protein to spread into the presynaptic cells. However, once in these cells, the spread gene is missing from the genome, and therefore the virus is stuck and can't spread into additional cells. And what we're left with is a unidirectional monosynaptic network. In my version of this system, each particle of the rabies virus carries in its genome a unique barcode, which is transcribed and can be captured during single-cell RNA sequencing analysis, along with the host cell mRNAs. So as a unique virus infects a starter cell, it then clonally replicates and spreads into the presynaptic cells. And we can use the presence of this shared barcode to define an infectivity path, which corresponds to the synaptic network. We then ID the starter cells from the presynaptic cells through mRNAs that functionalize those starter cells. And then by capturing thousands of host cell mRNAs from each cell in the network, we can simultaneously identify each cell's type and molecular state. So I named this system after my least favorite pizza chain restaurant, SPARO, which stands for Synaptic Barcode Analysis with a Retrograde Rabies Readout. Now, the size of SPARO datasets is critically limited by the diversity of rabies barcodes, and packaging rabies is a notoriously difficult and inefficient process. We therefore developed novel systems to achieve very high quality barcoding of DNA plasmids, achieving near theoretical levels of diversity, protocols to maintain this diversity through bacterial transformation, and finally overhauled systems for rabies virus packaging when the RNA virus is created from DNA that better preserve barcode diversity. And using my system now, we can generate libraries with millions of unique barcodes. Now, ideally, the abundance of each viral barcode in the infecting library would be predictive of the number of starter cell infections. So to test this idea and calibrate our libraries, I generated tens of thousands of starter cell infections under conditions with no viral spread, and then compared the number of starter cells infected to the abundance of genomic barcodes sequenced from the virus library. So you can see the results of that experiment in this plot, where each dot is a viral barcode. And as you can see, the number of infected cells here on the y-axis tracks nicely the abundance of the viral barcodes in the library, going from rare to most abundant. And in the most abundant cases, those viral barcodes are found in hundreds of cells. And with the rarest viral barcodes, they're found in essentially 1 or 0 cells. So these data provide a key part of the statistical framework we developed to infer synaptic networks based on the genomic abundances of individual viral barcodes. So to test this system, we developed a flexible system for culturing synaptic networks from dissociated embryonic mouse brains. And the system's tuned to incorporate cells from different brain regions or different mouse strains or even different species, and in which the number of starter cells can be easily manipulated in each culture well. Now, in vitro, monosynaptic networks have a real telltale anatomy with many presynaptic cells, seen here, spatially clustered around individual postsynaptic cells. And we designed our experiment such that in individual culture wells, we would have many of these discrete clustered groupings. Now, to sequence such networks, we dissociate the cells, fact sort to enrich for the GFP positive ones, and then we create single cell RNA sequencing libraries. We use the rabies barcodes to infer distinct networks and other cellular RNAs to identify cell types and whether the cells are pre or post synaptic. So this 2D embedding illustrates the molecular identities ascertained from about 150,000 SPARO libraries and in 150,000 uninfected cells from matched controls. Here, each dot is a cell, and neighboring cells have similar expression profiles, and the color code now reflects the basic cell class. So just to walk you through this briefly, growing in these culture dishes, we have 4 different flavors of glutamatergic neurons, shown here in red. We have neural precursor cells that are actively developing into astrocytes, and then 4 GABAergic lineages that include interneurons, spiny projection neurons from the striatum that we dissociated and co-cultured, and olfactory bulb populations from these caudal olfactory regions. There are also 2 major types of glia, or additional glia, polydendrocytes and oligodendrocytes. And I hope that you can appreciate by sampling thousands of mRNAs per cell, these SPAR libraries really exquisitely reveal cell types from diverse regions and dynamic molecular states as associated with development in vitro. Okay, so here I'm going to show you an example of a cell type specific monosynaptic network that we sequenced based on the expression of a shared rabies virus barcode. This one I'm cartooning up here in blue, which is extraordinarily rare in our viral library. So the blue barcode entered a postsynaptic medium spiny neuron and then clonally replicated and then spread into glutamatergic neurons, an additional spiny projection neuron, an astrocyte, and other neurons, etc. And a succinct way to illustrate the cell type composition of the presynaptic network is with a horizontal bar plot, such as this one. Of course, the real advantage of this system is that it's systematic and it's exceptionally high throughput, allowing us to quickly reconstruct thousands of synaptic networks. So here's a presynaptic network composition of over 9,000 such networks from these experiments in vitro. And really, if we focus on the top 100, the largest 100 networks, we can see that each one are made up of different combinations of many of the same cell types. Now, in vivo, we know that changes in membrane potential, i.e. a cell's firing patterns, tend to be cell type specific, and part of those signatures can be attributed to differences in the repertoire of presynaptic cell types. We therefore asked whether presynaptic network properties differ according to postsynaptic cell type during development. So here I'm showing one such difference that we discovered, which is the size of the presynaptic networks, as we compared between glutamatergic postsynaptic neurons and interneurons. And as you can appreciate, the interneurons tend to have, as a population, larger presynaptic networks. Now, to determine whether network sizes here are driven by a particular cell subtype, we can recode the data in a more granular way. So here, for example, I'm showing various subtypes of glutamatergic neurons and interneurons. And at this resolution, it's clear that the small network sizes in glutamatergic neurons are being driven in particular by this GRP expressing subtype. Now, these GRP expressing subtype glutamatergic neurons correspond to those from superficial layers, cortical pyramids. And the deep layer cortical pyramids tend to have larger presynaptic network sizes, more akin to the interneurons in the dish. And we know in vivo that the superficial pyramids tend to have smaller dendrites, and so that might account for some of these differences in presynaptic network size. So while the interneurons tend to have larger presynaptic networks as a whole, there's considerable variability in the presynaptic network size metric. And we sought to use this heterogeneity to identify candidate genes in postsynaptic cells whose RNAs might relate to presynaptic network size. And so for each cell subtype, we performed a differential expression analysis across postsynaptic cells with either small presynaptic networks or large presynaptic networks. And we asked whether any of our hit genes had known roles in controlling dendrite or synapse development. And we observed some interesting candidates. So one such gene was ARP21, which is an RNA binding protein, codes for an RNA binding protein, whose expression has been shown to positively regulate dendrite growth in vivo through knockout and ectopic overexpression studies. So one interpretation of these data is that postsynaptic interneurons that tend to have higher levels of ARP21 might have larger dendrites, which would provide more real estate for presynaptic innervation and hence larger total presynaptic network sizes. So I'm excited about these proof of concept results because they suggest we can identify molecules associated with particular network properties simply by measuring naturally occurring heterogeneity across thousands of cells during synaptic network development. And such results could be a great complement to population-level knockout studies from candidate genes and could help teach us about brain wiring in a more native context. So just to switch gears for the final part of the talk, rabies virus infectivity patterns have been used for decades to discover synaptic pathways in the brain, and yet a detailed molecular mechanistic understanding of how rabies virus crosses synapses and interacts with host cells is lacking. In our single cell data sets, rabies did not transmit to all neurons equally. Here, for example, I'm showing the developmental trajectory from neural precursor cells, NPCs, to mature spiny projection neurons, or SPNs. And as compared to the uninfected control cells in black, the rabies-infected cells in green tend to be enriched for the developmentally mature SPNs. So I use these differences to discover what molecular signatures were associated with enhanced rabies virus tropism. So to perform this analysis, I binned the cells along the trajectory from neural precursors at bin 1 to mature spiny projection neurons at bin 10. And I plotted the percentage of cells in each bin that were infected by rabies virus. And as you can see, starting around bin 5, there's a linear increase in rabies virus transmission. So there are 4 receptors hypothesized to mediate rabies virus entry. And to determine if receptor expression explains transmission, I plotted the normalized expression of NCAM1 RNA, which is the only candidate receptor gene with appreciable expression in our data sets. And as you can see, the onset of NCAM1 expression precedes the onset of increased rabies virus transmission that happens here, suggesting that merely expressing this particular receptor is not sufficient for transmission. To discover genes and molecular processes involved in rabies virus transmission, I identified genome-wide RNAs that correlated with this increase in rabies virus infectivity. And using this approach, I identified some 3,000 genes. And so to identify within this gene set cell biological processes that were enriched, I performed a gene ontology biological pathways analysis, and I identified pathways, 3 of the following pathways. 1, mitochondrial respiratory chain complex assembly. 2, phagosome maturation. And 3, positive regulation of synaptic transmission. And importantly, these associations were highly selective because thousands of control gene sets sampled at random or from expression matched spiny projection neurons failed to identify these pathways. And I was especially intrigued by number 3. So to specify which features of synaptic biology might be involved, I repeated the analysis using the synaptic gene ontology database. So while our rabies virus gene set was enriched for generic synaptic categories like synapse and presynapse and postsynapse, the only enriched very granular category was associated with the synaptic vesicle cycle. So here I am now going to plot the patterns of the correlated synaptical vesicle associated genes, which you can see nicely track with rabies virus transmission. So this was a hypothesis-free analysis which suggested rabies virus cell-to-cell transmission tends to co-occur in cells with molecularly mature synaptic function and offers us a list of candidate genes to further dissect this process. Okay, so to wrap up, in conclusion, single-cell, single-virion approaches like SPARO, I think, are poised to help evaluate wiring hypotheses by offering strategies for high-throughput characterization of molecular and synaptic properties of neural circuits. Because RNAs are captured in each cell, SPARO offers a flexible experimental system for studying synaptic networks in the context of molecular observation or highly parallelized manipulations, for example, CRISPR screens. And finally, genomics approaches can provide new insight into the details of viral tropism. So in my lab at the Vollum now, we're going to continue to develop SPARO, focusing on spatial transcriptomic methods to sequence synaptic networks now in vivo. So to illustrate how this works, we'll inject our barcoded rabies virus libraries into particular brain regions whose connectivity we want to map. We'll cut serial sections and array them to spatial transcriptomic devices, such as SlideSeq. And then using these spatially resolved data sets, we'll capture multimodal information about each cell, including the spatial locations of cells, discrete synaptic networks based on the shared viral barcodes, and then using the host cell RNAs, identify the cell types, expression states, and, for example, human genetic variants that we can overexpress in the system. So this is just one of many potential use cases of the technology. And finally, I'll close that our lab is really interested in working out many of the unknown features of rabies virus biology. And these include identifying their entry receptors, host factors that mediate rabies virus interactions, and then mapping the genomic interactions between virus and host cell and how that stratifies across cell type. So with that, I will conclude and thank my mentor, Steve McCarroll, and the McCarroll Lab and the great team we assembled to work on Sparrow, and especially our collaborators in Bernardo Sabatini's lab, Kiwi and Addy, who helped us with the viral packaging protocols. And just to plug my own lab here at the Vollum, so we're building a real transdisciplinary team of virologists, computational biologists, and neuroscientists to study viral behaviors in neural circuits with the hopes of creating better viral vector tools for brain mapping. So with that, I will close and take any questions. Thank you very much.
Okay, thanks, RP. John, you raised your hand.
Oh, thanks. Thanks, RP. That was really, really cool. I have a technical question that is at best perhaps esoteric and maybe at worst irrelevant, but I'll ask it anyway.
Perfect.
Yeah, right. So in terms of representation of connections, in terms of picking up the barcodes, I imagine these are expressed at a high enough level you don't worry about dropouts, or do you worry about dropouts?
Well, that's one advantage of rabies virus is that, you know, on average, these barcoded or the barcoded RNAs make up 3 or 4% of all of the RNAs in the cell. So they have very high expression levels. So certainly for smaller libraries, dropout is a thing, but our experiments suggest that ascertaining the viral barcodes is both like very sensitive and selective.
Cool. So otherwise, you think you're getting a pretty even representation of connections of the types of cells that might be forming the presynaptic context? Yeah,
I think that's kind of presumptuous for me to answer yes. I mean, we just know very little about the molecular biology and cell biology of how the virus moves across synapses. There's my kind of, I think the short and sweet of it is essentially like you need to have receptors and membrane turnover. And synapses are great sites of those things. So we're definitely interested in understanding how synaptic properties like membrane turnover, presynaptic release, those kind of meta properties of synapses map onto the probability that a virus will move from a dendrite to an axon. And hopefully the goal is to generate data sets that are large enough that you can identify even low probability events and then kind of study the cell biology of those in addition to the high probability events.
Yeah, I mean, presumably you have a way of validating this, right? I mean, you should be able to benchmark this in some way.
Yeah, we're starting with electrophysiological analyses just to kind of track which cells that the rabies goes into. And, it's complicated, like cells, there's striking innate immune responses that have kind of temporal kinetics to them, and those need to be understood as well. So those are kind of key things that we're working on now.
Great, thanks.
Thank you. Hold on a second, there's 2 questions on the chat board. So first, Ning, Tianning, do you want to ask a question yourself?
Oh, yes, I can ask.
Okay.
Can you hear me?
Yes.
Yeah, go ahead.
Okay, so the rabies has been used quite a while for labeling supposedly synaptic connected cells. Do you know anyone actually really confirmed all the rabies labeled cells are actually functionally coupled or part of them are coupled? Any information about that?
So yeah, I mean, I've scoured the literature for really comprehensive and systematic analysis of that, and I don't think it's been done. I think there's reasonably strong anecdotal evidence in the sense that rabies virus presynaptic spread patterns tends to kind of fall into presynaptic cells that correlate with postsynaptic responses, even in contexts when the anatomy of the postsynaptic cells are really highly intermixed. But those experiments are challenging to do, and we're setting out to do them in a variety of ways with both minimal cell biological synapse systems and electrophysiological investigations.
So you also mentioned that the rabies coupling seems varies on different cell types. Do you think maybe cells with different type of synaptic connection also have different preference by labeled by rabies? Will that be a possibility also?
Yeah, I mean that's an unknown. I think it depends on what receptors that the rabies virus uses and the membrane turnover at those synapses. And so, for example, whether basket cell synapses on the soma, which are right next to the special organelles that rabies virus makes to replicate its genome, whether those are more accessible to very distal synapses in the dendrites, it's plausible that that could be the case. I mean, I guess the hope is that we can attempt to study these features of synaptic transmission in the virus. And once we kind of get a sense of the cell biology, then generating the very high throughput data sets will have some more kind of meaning and meat to them. That's the goal. And I should say that the technique is not supposed to be like a replacement for EM connectomics or electrophysiology or really anything. It's just supposed to generate a different type of more aerial descriptions of probabilities of connections that can help guide us to key synapses among certain cell types that might be relevant in disease model contexts or just during regular development.
I guess what I meant is, could that rabies pass exciter and inhibitory synapse in a different rate or different way? So even you map this and you get like a partial or biased labeling. Is that possible?
Yeah, I mean, I think that's definitely true. I mean, the type of connectivity mapping is binary. It's digital, right? Connections are not. And if excitatory synapses have a lower probability of transmission than inhibitory synapses, and we record thousands of instances of monosynaptic networks, we hope we can kind of detect those phenomena.
Okay, I have moved on to one last question from Gang with his hand. Do you want to ask a question yourself, Gang? Gang? I see your hand.
Okay, can you hear me, Zilong?
Yeah, please.
Okay, thank you. So very beautiful work. So I have a question about this the called triangle problem. Since the starter cells, all of the starter cells have G proteins, so they can be complemented. But if the starter cells connect with each other, so there is a possibility that the virus contains a specific barcode can move from one starter to another starter and then go to the upstream circuit. So there is a possibility that all the labeled all the barcode can be direct and indirect upstream circuit. So can you differentiate those direct and indirect barcode?
So that's a great point, right? If a starter cell is part of another starter cell's presynaptic network, then the virus in principle could kind of hop through that starter cell as well. I think I have two answers to that. I mean, in the experiments that we set up, we purposefully made the monosynaptic networks to be very sparse in the dish to try to avoid that problem. So we just controlled the knob essentially to make the starter cells and their networks relatively non-overlapping. But I think it's a really important point. And now we're working on simply making inducible systems that we can control glycoprotein expression and track starter cells with different vectors so that we can just kind of control the window of time when the glycoprotein is around in the cells to complement the virus.
Okay, thank you.
Okay, so I have a few more questions on the chat board, and also I will type my question in chat board. Thanks. Aaron, would you move to Holly to talk?
Okay. Thanks, RP. Awesome.
Thank you all.
I'm sure you're going to be getting a lot of postdoc applications. If I was still applying for postdocs, I would apply to your lab. So it's my actually, it seems like today's theme is speakers who went to college at suburban Philadelphia liberal arts colleges. RP went to Swarthmore, and our next speaker, Holly Klein, went to Bryn Mawr College. So it's a great pleasure to welcome Dr. Holly Klein as our next speaker. She's the Hahn Professor of Neuroscience and the Chair of Neuroscience, Chair of the Neuroscience Department at Scripps in California. And after Bryn Mawr, Holly did her PhD at UC Berkeley, and then postdoctoral fellowships at Yale with Martha Constantine Patton, and then at Stanford briefly with Dick Chen. She then started her own laboratory at the University of Iowa, and then moved to Cold Spring Harbor, where she was, including director of the laboratory until 2008, when she moved to Scripps in San Diego. Holly's won many awards. She's been the past president of the Society for Neuroscience. And the goal of her research lab and her focus over the years has been to define the mechanisms by which sensory experience affects brain development, form, and function. She's pioneered the retinotactile system of frogs and done wholesale recording to study activity-dependent plasticity in the visual system. She's identified activity-dependent mechanisms to control plasticity of dendrites and axons. She's studied synaptic maturation and map formation, and discovered roles for protein kinase activity in generating such spatial maps. And then her overarching impact of her work is that she's demonstrated using multiple approaches that circuit formation during development is a dynamic process and is refined based on experience. So really looking forward to hearing the latest from Holly. And it's an interesting thing in science where you can be starstruck by the legends in the field and then get to know them and have them as your colleagues. So it's great to have Holly here as a speaker. Thanks for joining us.
Yeah, thank you very much, Aaron. And I'd like to thank you again for organizing this incredible seminar series. It's really been, I'd have to say, uplifting during the last year. So thank you for that. I'm going to talk about some of the work that we've been doing and thinking about on the potential role of extracellular vesicles in brain development and function. So we know from a variety of many studies that intercellular communication is required for brain development and function. And a lot of these kinds of intercellular interactions have to do with cell-cell contact or release of neurotrophins or mechanisms like that. But there are several studies that sort of woke me up to this amazing possibility that extracellular vesicles might have signaling functions in the brain. And one of the, one series of studies that really struck me came from Zandra Breakfield's lab along with her colleagues, where she showed that glioblastoma release extracellular vesicles or exosomes that then, depending on the cell type that these vesicles contact, they mediate a variety of different effects in the tumor environment, including vascularization and metastatic niche formation and immune response modulation. So it seemed like anything that goes wrong in disease or that is used as a fundamental mechanism in disease might also be operating in the healthy brain. So that struck my fancy. And then there was another series of papers that came out of Vivian Budnick's lab. She works on Drosophila, and what she did was to show, using a variety of genetic tools, as well as anatomy and cell biology, that exosomes function during the development of the neuromuscular junction in Drosophila, where the motor neuron axons release exosomes, which then signal to the muscle fiber and are required for initiating formation of the neuromuscular junction. So these series of papers really caught my attention. So extracellular vesicles are released from basically every cell in our body and in the body of most or all other animals, as far as we know. Extracellular vesicles are heterogeneous and can be described most easily by their physical properties, but also their pathway of biogenesis. So I'm going to talk about exosomes. They're generated through a pathway that involves a multivesicular body, And the multivesicular body then fuses with the plasma membrane, releasing the extracellular vesicles or exosomes into the extracellular space, where they can then contact other cells in the environment. So in the brain, again, virtually every cell type that we know about releases exosomes. And it's quite interesting to think that all of the different components within the brain can signal to other neural cells and receive signals from them. So for instance, astrocytes send signals to neurons and receive them, vice versa with microglia, as well as other neural cell types. So this was fertile ground for thinking about how exosomes might influence brain development and function. So we dipped our toe into the water first by isolating exosomes from human iPS cell-derived neuronal cultures. And we could isolate the exosomes by a series of centrifugations and demonstrate their purity according to transmission EM and biochemical features, which are shown here. In the first set of experiments, we asked whether or not exosomes that we harvested from the conditioned media of human iPS cell-derived neurons could promote neurogenesis. So we took these exosomes and delivered them to human primary neural cultures over a period of several days, after which we fixed and analyzed the cells. And what you see here is by labeling the cultures with DAPI and MAP2, we saw an increase in the total number of cells and also in the MAP2 positive cells within the culture over a period of several days. So that was encouraging as an initial experiment. In addition, we wanted to test whether or not exosomes have neurogenic bioactivity in vivo. And the way that we did this was to harvest exosomes from primary rat neurons. We established the neuronal culture from the E18 cortex, collected exosomes and treated some of them with protease K to inactivate them and left the others untreated. We took these two populations of exosomes and injected them into the lateral ventricle of P4 mouse pups. And then the next day, we analyzed whether or not we could see a change in cell proliferation within the brain. And the area that we chose to look at is the hippocampus, which is very well known for neurogenesis in the dentate gyrus. Here are the images from a representative mouse brain treated with the proteinase K treated exosomes or the untreated control exosomes. EDU is labeled in pink here, and you can see very clearly in these examples that there's more EDU labeling in the dentate gyrus of the control exosome group than the inactivated exosome group. And when we look at higher power and we count them in an unbiased manner, we see that there's a significant increase in EDU labeling in response to exosome injection into the ventricles. You can also see a ton of proliferation in the cortex, but we didn't count it there. So this suggests that by ventricular delivery of exosomes, they can actually penetrate and have bioactivity within the developing brain. We were interested in learning a little bit more about how exosomes might signal and influence brain development in a neurodevelopmental disorder. And so we started to collaborate with Alison Wertree, who has done a lot of work with Rett syndrome, as has Huda Zavid, who wrote the review that I'm summarizing here. Rett syndrome is the result of the loss of MECP2, which is methyl CPT binding protein. This results in large-scale aberrant gene expression. And we were imagining that this would predict large-scale changes in exosome protein cargo. So that's why we chose this type of neurodevelopmental disease to pursue. Alison Moertry's group had demonstrated previously that neuronal cultures established from Rett syndrome patients had aberrant electrophysiological responses, and you see here a decrease in mini amplitude and frequency in the cultures from Rett syndrome patients versus the isogenic controls. And similarly, there's a decrease in network activity shown here, looking at calcium imaging in wild type cultures versus the Rett cultures. So what we did was collect exosomes from the Rett syndrome neural cultures and from the isogenic control neural cultures, and analyze those exosomes using proteomics. And this was in collaboration with John Yates here at Scripps. And so we were happy to see that there are about 2,500 proteins in neural exosomes, 750 of which were annotated to have neural functions, and about 240 were differentially expressed between the two populations, the two sources of exosomes. And I'm just going to quickly show you a bit of the analysis of the bioinformatics. Looking at the gene ontology categories, we see that many of the categories that are statistically different between the two populations of exosome proteins concerned neurogenesis and nervous system development. And if we look at the same type of analysis, we specifically see a variety of different categories that pertain to development of neurons, axon guidance, dendrite development, and these other features that are near and dear to people who are interested in mechanisms underlying brain development. Furthermore, we noticed that if we took the two data sets that are categorized according to neuronal proliferation or neuronal development, we found using string analysis that many of these individual components function within a network. And so it was attractive to think that maybe what exosomes do is they basically package together all of these components of signaling networks and deliver them to cells to sort of boost a kind of proliferation or neuronal differentiation process. So we were interested in collecting exosomes from either the control cultures or the Rett cultures and testing their activity, their bioactivity in recipient cultures. And the types of bioactivity that we thought would be interesting and relevant are proliferation, neurogenesis, differentiation, synaptogenesis, and circuit function. And I'm going to tell you about our experiments exploring these outcomes. So first we found by introducing control exosomes to control human cultures that the control exosomes increase cell proliferation. So the protocol is shown here. We have our primary neural cultures. We add either control exosomes or Rett exosomes a couple times. We pulse the culture with EDU, and then we do a variety of different labeling techniques. And what you see here is that the isogenic control exosomes increase EDU labeling, increase cell proliferation within the cultures, whereas the MECP2 derived exosomes do not have that bioactivity. When we label these cultures with double coordinate to test whether or not these newly generated cells differentiate into neurons, what we find is that many more of the newly generated cells differentiate into neurons in the cultures exposed to the control exosomes compared to the MECP2 exosomes. By contrast, when we label the cultures with GFAP to evaluate the development of astrocytes within these cultures, we find that exosomes from either the control cultures or the MECP2 cultures are equally capable of driving astrocyte development in these cultures. And so this indicates that the isogenic control exosomes have, and also the RET exosomes, both have bioactivity in this context, but the bioactivities differ between the two. So we were then interested in testing whether or not the recipient cells modify or control the degree of bioactivity that the exosomes have. So you can imagine if we introduce exosomes to one kind of recipient cell, they may be capable of responding to that signal or not. And again, we were interested in the same range of outcome indicators as I had mentioned before. So here, again, what we did was we had human primary neuronal cultures. We either knocked down MECP2 or had a control knockdown, and then we introduced the control, in this initial experiment, the control exosomes. What we found from this experiment was really quite exciting. In the absence of exosomes, there's relatively little EDU labeling here, but we found that the proliferation deficit that's present in the control conditions with MECP2 knockdown is very well rescued by introduction of wild-type exosomes, even to a greater extent than IGF, which is typically used as means of counteracting the effect of the RET deficits. Similarly, the wild-type exosomes were able to rescue deficits in cell differentiation, in neuronal differentiation in the knockdown culture. So this was a very exciting result for us. And so it encouraged us to look a little bit further to test whether or not wild-type exosomes can rescue synaptogenesis and neuronal activity in the RET neural cultures. So here, our cultures are the iPS cell-derived cultures from the RET patients. We grew these for a number of days, added exosomes from the wild-type cells, and under these conditions, we labeled the cultures with MAP2 and with synapsin to identify presynaptic puncta. And you can see here that there's a significant increase in synapse formation as a result of introducing the exosomes to these RET cultures. And then finally, in this series of experiments, what we did was test whether or not the wild-type exosomes could rescue or improve network activity in neurospheres generated from the RET patient iPS cells. So we established the neurospheres and lay them on top of a multi-electrode array, and then we introduced the wild-type exosomes. What you see here on the top are the results of the multi-electrode array recordings when we added the RET exosomes. So one thing to notice is that there are relatively few electrodes that registered any activity at all under these conditions. And when there was activity, it was relatively sparse. But in neurospheres to which we added the wild-type exosomes, first off, we found many more electrodes that registered activity, and many electrodes had a lot of continuous activity over time. And then the other interesting observation is that there was a lot of coordinated activity across neurospheres and across electrodes in the neurosphere. So these data indicated that exosomes have neurogenic bioactivity, both in vivo and in vitro, that the exosome proteomics indicate that neuronal exosomal cargo include components of signaling networks that play roles in neuronal development. When we then deliver exosomes to cultures, either wild type or the RET cultures, we find an increase in cell proliferation, neuronal differentiation, synaptogenesis and circuit activity. And we find that the wild type exosomes have bioactivity that rescues the neurodevelopmental deficits that we see in the MECP2 loss of function neural cultures and neurospheres. So one of the reasons why I like this set of experiments is that basically in doing this, we worked out a platform which allows us to analyze disease genotype effects on exosome-mediated intercellular signaling. And so I'm just going to run through this. This is basically the experimental design that we use, but I just want to elaborate a little bit more. So what do we need to know to understand how exosomes might be signaling to recipient cells? We need to understand what the cell type specific cargo are. I anticipate that the exosome cargo will change according to the state of the cell. So for instance, we already know from experiments that we've done more recently that exosome cargo vary depending on the age of the neural cultures. And we also expect that it changes in terms of activity state. The other thing to understand is about the recipient cells. I anticipate, again, that the responses of recipient cells will depend on the cell type and the cell state, its point in development or aging, and of course, its genotype. And so one scenario is you establish cell type specific cultures from either the disease genotype or the CRISPR corrected control cultures. And then you can collect these cell type specific exosomes for analysis, not just by proteomics, but also by mass spec and other kind of lipidomic kind of analyses. And one advantage that will come out of this, detailed information about the exosome markers, is that we will be able to take better advantage of patient samples, for instance, from blood or other liquid sources to use as biomarkers. So then the next phase of the experiment that, again, I described above was to take cell type specific exosomes from different genotypes or control corresponding controls and test their bioactivity in a cell type specific manner, and then reverse to take the cell type specific exosomes and compare them across the genotypes and across cell types. And I feel like this kind of strategy will really allow us to systematically evaluate the potential role of exosomes in intercellular signaling across ages and stages and cell types and disease genotypes. And with that, I'd like to thank the people in my lab who've worked with me on these projects. This is Pranav Sharma, who was drove the RET project. This is Lorraine Caponati and Anna Bardusco, who are currently working on extensions of this project, and I'm happy to acknowledge my collaborators and funding. So thank you very much.
Thanks so much, Holly. That was really awesome and exciting. I had a question or two. You mentioned you want to figure out the cell type and cell state differences in exosomes, but I imagine within a single cell in a single state that they are using exosomes for lots of different things, right? They are probably, so there might be, even within a cell state and cell type, they probably would need a way of saying, I want this cargo, but not this one. I want proteins, I want RNAs, maybe. There might be RNAs that are differentially packaged. So I'm just wondering like what your current thinking of how like that neuron or type knows which signaling molecules to put into exosomes at which time.
Yeah, so the biogenesis of the exosomes is really fascinating, gets deep into a lot of cell biology and still is too I guess, doesn't give us the spatial and temporal information that we need to be able to answer your question. However, one strategy that we used was, this is basically why we used human iPS cells, because you can grow a lot of them. They can be relatively similar across cultures if you're careful in your differentiation. And then the other thing is that we haven't done this, but it certainly is possible to use something like a Campano chamber to basically collect exosomes that might be released from different parts of a cell, let's say an axon or a dendrite. And so I'm imagining that would be feasible and might get close to answering your question.
Is there like a genetic way of eliminating exosomes, or like to prevent them from being produced?
Well, we've been working on trying to find a way to block their release, because I mean, my gut reaction is that I think they do so many things for the cell that to just sort of block their biogenesis wholesale ends up with a dead cell. And for instance, I mean, some of the biogenesis pathways, if you block them, the cells just don't live. So I think we need something a little bit more nuanced.
Got it. Okay. Scene.
Oh, Holly, wonderful talk. So is there a genetic or immunostaining-based way to see the genesis happening, like exiting the cells, like in addition, I mean, especially in vivo, right? Is there a way to see these things really happening, or is really just biochemical is the only standard?
Well, so now there are a number of reporters that have been generated where you can basically tag the vesicles when they're still inside their host cell or their generating cell with, for instance, with GFP. And this is actually great because then you can observe the release of the exosomes and try to track them. So that's pretty tricky in terms of microscopy, but there are reagents out there that can do that. And
related to that is me, since all of us study the neurobiology. So from your perspective, you see a dendritic axonal bias of this exosome versus a stomata general release, like any other cells? Like any specialized structures that may be accountable for this, or it's just general release? Is there an axonal enriched release machinery?
Okay. Oh, so actually, Chris Walsh and Wei-Chung Lee published a paper a few years ago where what Wei did was use his serial section through the cortex and specifically look for the distribution of multivesicular bodies throughout neurons and in different layers and stuff. And it was a really astounding demonstration of how widespread multivesicular bodies are within neurons. They are certainly in axons. They're in, you can find them in, close to boutons. They're all over dendrites. So I think this is directly relevant to Aaron's initial question, is that you would anticipate that they would be packaging different cargo for region-specific signaling. And so we have, we've labeled specific exosome cargo, mostly to see how where they go in the postsynaptic cell. We didn't actually use it in the way that you've been thinking in terms of where in the cell are different cargoes assembled and released. But it's a very interesting question.
Thank you.
So Holly, the rescue of the Rett phenotype or of the activity was really striking. Do you have a sense, like from dilutions, like how many exosomes you need? Like, I'm sure one exosome is too few, but do you have a sense of how many exosomes will it take to rescue this, or is it dose dependent?
So I can say that it's dose dependent. So what we did was we harvested the exosomes, we measured the amount of protein from which we can estimate the number of exosomes, and then every experiment that we did, we introduced 3 different dilutions. And it was very interesting because in some cases, some bioactivity was present at one dilution, but not others, and vice versa, depending on what the outcome measure was that we were looking at. And so that's interesting in terms of trying to then, narrow down the scope to see what cargo are causing what outcome measures. So I have to say, I've read papers where they say, we're introducing the equivalent of 2 exosomes per neuron and they see an effect. I'm so skeptical about that. But at the same time, I'm reluctant to give you a number of how, what our exosome to neuron ratio is.
Got it. RP, did you want to ask your question that you put on the chat?
Hey, thanks for the great talk, Holly. I wanted to ask about exosome functions in viral mediated signaling in the brain. And if you know anything, if you can give us maybe a sense of whether exosomes can be co-opted by viruses to move RNA around, or whether innate immune molecules move around through exosomes, or what's the backdrop on that?
So Jason Shepard has pretty strong opinions about exosomes and viral and mRNA particles moving around together. When we were trying to understand the mechanisms of exosome uptake and whether it was bidirectional, it seemed like there was a lot of potential overlap between how viruses enter cells and in terms of the cell biology. And actually that was something that I was thinking about with your talk, just about what kind of receptors and proteins are present at the synaptic face that would allow viral transfer, rabies virus transfer. And I unencumbered by data, I think that there will be a lot of overlap between these cellular mechanisms that relate to uptake of exosomes and uptake of viral particles. So another comment is that we do see immune molecules in exosomes. There are all kinds of interesting things in there. So.
Yeah, I just think it's interesting, like what the valence of that type of signaling would be for infection, right? Is that something that can prime a cell from the viral perspective to be infected, or is it kind of a defensive posturing from one cell to another when the first cell has been infected? I don't know. It's just it's
right, but you would have to pose that question in a way where you knew exactly what the history of that cell was, so that you could say, I want it to have been, alert to infection, or, so you can't just take any old cell and dump the exosomes on them and get the answer that to the question that you're interested in.
Well,
right?
if you want to profile some exosomes after rabies virus infection, and then we can do some titering, that would be pretty cool.
Okay. Thank you.
Ying Liu has a question about if the components of exosomes are influenced by neuronal activity.
Yes, they are. So there are a few activities really interesting. So first off, it increases release of exosomes. We have some evidence that it modifies the cargo in exosomes. And then the other interesting part that we haven't specifically tested is, are active or quiescent cells, do they respond differently to exosomes, right? Wow. So I feel like I bring this up because, you can't we have to consider all of these partners in the signaling process.
It must be all this amazing sorting machinery that is left to be discovered.
Yeah. So one of the, for you, Aaron. So of course, well, I don't know if this is an of course, but the synucleins, alpha and beta synuclein, are both packaged into exosomes, and the other thing that we can say is that the efficiency with which different cargo are packaged varies on the cargo. So for instance, the synucleins are like supercargo, but other proteins are less well packaged.
Wow, amazing. It seems like Kai Wen had a question.
Yes. Yes. Thanks, Hollis, for a great talk. And my question is similar to. Oh, can you hear me?
Yes.
Oh, yes. So it kind of in line with the last one. So my question is, how is the exosomes release and reuptake coordinate with the other intracellular events in either neurons or astrocyte, depends on who's the host, right? Generate these cargos. And second of all is the signaling mediated by the exosome. Do you think is the exosome per se is important, or just the cargos that been held within the exosomes is important? I mean, if you exchange the exosome with just the cargos inside, will that also serve the purpose of this exosome transmission and reuptake?
So I'll answer the second question first because I can still remember it. So that's super interesting because implicit in your question is that the lipid protein components of the exosome membrane are themselves capable of signaling. And so we know that exosomes have bioactive lipids, and I presume those are embedded in the membrane, which means that they would signal maybe completely independently of the cargo, of the other cargo, the protein cargo, right? So again, I feel like these are super interesting experiments that need a very careful, intentional design. I don't think we're going to stumble across a result that would answer that question.
Yes.
So then your other question was,
how is the exosome signaling coordinate with the intracellular signaling of the host cells, such as the neurons and the astrocytes, which is kind of in line with the previous questions, how the neural activity may regulate the exosomes transmission and reuptake, or if it is released by the astrocyte, how is the astrocyte I just don't know. It feels like it's two pathways of activities, right? One is extracellular, and the other is intracellular. How are these two pathways coordinate with each other? So do they work independent of each other, or any direct link with each other?
Yeah, I think that's super interesting. So you can imagine, let's say there's a number of potential recipient cells, and you have a flood of exosomes that are released, and the exosome can actually enter into all of those potential recipient cells.
Yeah.
But the only recipient cells that can respond to those exosome cargo are ones that, I don't know, recently had MAP kinase signaling, or some specific trigger in the recipient cell that would be a gate for whether or not it can detect and respond to the message that's coming from the exosome. So that's why I feel like, that's a little too chaotic. If this is going to work, there has to be a little bit more consistency in the opportunity for signaling. But I feel like now if we go and plan in vitro experiments that would specifically test these kinds of questions, we could address them. Like activate all the cells and see, does that increase the probability of having, a signal transferred, right?
Yes. Yes, yes, indeed. Yeah, very interesting. Thank you.
Thank you.
Okay. Super exciting and provocative. I don't think we have other questions. Okay, thanks so much, Holly, and thanks, RP, of course. Thanks everyone for joining.
Great. Thank you very much, everybody.
All right, thank you.
Yeah,
Holly,
see everyone next
great.
week.
Yeah.
See you next week.
Bye.
Great talk.